Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
The human immune system is a highly complex network of cells, signals, and responses that is tightly regulated to ensure that the body can fight off infection without damaging its own tissues. Now, researchers from Japan report a new way in which the immune system protects lung tissue from viral infections. In a study published in Cell Reports, researchers from Nara Institute of Science and Technology, NAIST, have revealed that antigen-specific killer T cells, CD8 T cells, rapidly expand in the lungs when they encounter antigen-presenting alveolar macrophages, AMs, to protect against viral infection. CD8 T cells confer protective immunity against infection with respiratory viruses, such as influenza virus, IAV, and severe acute respiratory syndrome coronavirus 2, SARS-CoV-2, by killing infected cells. In order to target the correct cells for killing, naive CD8 T cells must be primed by contact with antigen-presenting cells, APCs, which mediate the uptake of virus-infected cells and present their antigens, in a process known as cross-presentation. The primed CD8 T cells then clonally expand and differentiate into effector or long-lived antigen-specific memory T cells. Multiple cell types can present antigen to CD8 T cells in the lungs, although the role of tissue-resident macrophages in this process is unclear, explains Takumi Kawasaki, lead author of the study. AMs are the first cells in the lungs that encounter infectious materials, environmental particles, surfactants, and dying cells, and they are important for the host defense against bacterial and fungal infection, so we suspected that they were also important in protecting against respiratory virus. First, mice were primed by vaccination with a specific antigen or infection with IAV, and then they were subjected to secondary immunization or reinfection. We determined that antigen-presenting AMs present inhaled antigen to memory CD8 T cells, says senior author of the study, Taro Kawai, and that this resulted in a rapid expansion of antigen-specific CD8 T cells in the lungs. Furthermore, the researchers found that AMs help to develop resident. Importantly, administration of antigen-loaded AMs to mice induced the proliferation of resident memory-type CD8 T cells. This strategy may improve the efficacy of CD8 T cell-dependent cellular immunity, says Kawai. Given that the lung is a major tissue for IAV and SARS-CoV-2 infection, the findings from this study regarding the mechanism of lung-resident memory CD8 cell expansion are expected to lead to the development of new vaccines that induce cellular immunity. Virus-specific antigen-presenting AMs could be delivered as a type of cell transplant vaccine in the future. Make a difference. Sponsored Opportunity StorySource. Materials provided by Nara Institute of Science and Technology. Note: content may be edited for style and length. Journal reference: Takumi Kawasaki, Mo Ikugawa, Kosuke Yunoki, Hifumi Otani, Daisuke Ori, Kenjei Ishii, Etsushi Kuroda, Shiki Takamura, Masahiro Kitabatake, Toshihiro Ito, Ayako Isatani, Taro Kawai. Alveolar macrophages instruct CD8 T cell expansion by antigen cross-presentation in lung. Cell Reports, 2022, 41, 11, 111828 doi, 10. 1016 J. CellRep. 2022. 111828 Cite this page. Nara Institute of Science and Technology. Alveolar macrophages help CD8 T cells go anti-viral. Science Daily, the 28th of December 2022. ift.tt/ogweek. Nara Institute of Science and Technology. 2022, December 28. Alveolar macrophages help CD8 plus T cells go anti-viral. Science Daily. Retrieved December 28, 2022 from ift.tt/ogweek. Nara Institute of Science and Technology. Alveolar macrophages help CD8 plus T cells go anti-viral. Science Daily. ift.tt/ogweek. Access December 28, 2022. Thanks for watching. Please subscribe my channel for more videos.